So, hello everybody around the globe, and thank you for joining our Hopsteiner Forum. Today we are diving into the fascinating world of product development guided by scientific studies. My name is Christina Schmidt. I'm the head of special analysis and sensory, and it's my pleasure to be your host for today's discussion. For those of you who are joining us for the first time, I'd like to give a brief introduction to our webinar. So the webinar is a free online seminar with presentations covering a range of hop-related topics. Through the sessions, we aim to keep you informed about the latest research in hops. Whether we are an expert or just beginning to explore the world of hops, we're here to share valuable insights and discoveries with you. Before we start the presentation, I would like to point out that I will be happy to receive your questions and comments. Your microphone and camera remain switched off during the webinar, but you can address your questions at any time through the Q&A tab in the control panel. We will then pick up your questions at the end of the presentation. The presentation will take about 30 to 35 minutes, followed by the mentioned Q&A session. So today's presentation will be held by Dr. Alexander Feiner, Head of Innovation and Research at Hopsteiner. Throughout today's session, we will be delving into scientific studies and product development in the world of hops. Alex grew up on a hop farm in Hallertau and studied agricultural management at the University of Applied Sciences Weinstefan in Germany. After joining a Hopsteiner research program in Yakima in the US as an intern, he finished his Master of Science in 2012 and his doctoral thesis on hop genetics and metabolomics in 2020. Alex joined our Hopsteiner team in 2012 and is nowadays leading innovation and research at Hopsteiner in Mainburg. So Alex, we are curious about more insights of your work. The stage is yours, and to guide us through your findings.
Thank you. Yeah, thank you very much, Christina, for your kind introduction. And yeah, welcome everybody to our today's seminar. I'm very proud and very happy that so many are joining today. And as Christina said, yeah, my presentation today is about product development and how scientific studies and scientific work is influencing that kind of development. First, I would like to start with a soft introduction about a few global facts about hops. What are we talking about? We are talking about a dioecious crop. So there is a male and a female plant. This is the female, the cones, everybody knows, and the male plant. And it's perennial, so it grows every year. And there are fields out there which have been cultivated for decades. It's diploid, so it has 2 × 10 chromosomes. And when we talk about hops, we talk about the species Humulus lupulus. There is Humulus scandens and yunonensis beside, but the one which is of commercial interest is Humulus lupulus. And worldwide are around about 60,000 hectares cultivated of that, with US and Germany are the main growing areas, covering about 75% of the global hop production. And nowadays, currently, more than 95% of the hop production goes into the beer production. So the female flowers, the cones, are used to give aroma and bitterness into beer. There is also pharmaceutical applications or animal feed applications, but currently, as I said, more than 95% goes into beer. And what is driving our business, and what is one of the major drivers is the global climate change. It's as I said, it's global. Since the 1950s to 80s, we have more than a degree of Celsius increase of temperature. And talking about especially Europe, it's not 1, it's more like 3 degrees higher than it used to be. And this has a heavy effect on the Arctic ice. It's melting, but also sea level is rising. And one major driver is carbon dioxide, but I don't want to discuss today what is causing the global climate change. It is just the fact that it's present and it's really influencing our daily business and the hop cultivation. As you see here, For example, in Yakima, in the US in summer 2021, there were temperatures above 45 degrees Celsius, and this is actually the time where hops need most of the water. And even with drip irrigation, some varieties and some plants were not able to get enough water into the top of the plants, and so they started to die and to wilt. It was a good year for us in the research and breeding program to select certain genotypes, but it was definitely a bad year for hop cultivation. And the last 2 years in Germany, 2022 and 2023, we had during summer really high temperatures, higher than average, and also a very different and changing distribution of precipitation. And during the months of June, July, hops need a lot of water, and if that time temperatures are high and precipitation is low, yeah, you expect low yields, and this was then actually also the effect in 2022 and 2023. But for that we have we try to create solutions, and we already have created solutions. We have a Hopsteiner research program, a global one. One is one station in the US and in Yakima, where the breeding program started in the late 90s, with nowadays the focus on high alpha and the craft beer market. And in 2012, we started the breeding program also in Europe, with nowadays the focus on aroma and high alpha. And here in Mainburg, we have the breeding location and breeding varieties also for the European growing areas. And as I said, climate change is something which influences our business, but also regulations like pesticide regulations, such as European Green Deal and so on. So what we are looking into is really disease-resistant and drought-resistant varieties. So this is a really important trait in hop breeding, but also yield stability under less input of resources like fertilizer, like pesticides, but also under drought conditions, we need stable yields and also a certain chemical profile. Because at the end, all what we want is a new hop variety, which is of course resistant, but also gives a pleasant bitterness quality and aroma quality. Because as I said, most of the hops are used in brewing, and therefore, the future varieties, they need to be as good as the old ones in terms of bitterness and aroma quality, because we want to make beer better in the future. And every year we start with a certain amount of crosses. So we use a female plant as a mother parent and a male plant as a father parent. And then after 5 years, we see the first yields and the first chemical profiles in the mature state. And after 5 years, we have an idea if the crosses we made were successful or not. And the best genotypes are then advanced into in acreage, and then we get enough material out of that for major brewing trials. And then after nearly 10 years, we find out if we were successful and if we have developed a potential new variety. So that takes very, very long time and is very time consuming. And therefore, what we do at Hopsteiner is, we try to elucidate complex traits like aroma, like resistance, like yield, and we look deeply into the genomics, so what can basically happen, and also on the metabolomic side, so what actually happens in the plant. And as my title of today's presentation is Science-Guided Product Development, now I would like to give you an overview what that actually means for us. And because it's not only breeding, it's also making the next generation products out of it. So what we do, we look into the hops, into the raw hops profile, and we try to screen the best out of it, try to screen them with genetic markers, with chemical markers, and so on. And we also look into beer. And what we try to find is, or what we try is to identify aroma relevant compounds in hops and their role in beer. So what is potentially in a hop plant and what ends in beer? And this gives us then results for the breeding program. So to make the right varieties with the compounds we need, but also then afterwards develop the products out of it. What makes then yeah, the future products at the end. And at the end, what all this means is, yeah, with new innovative hop products, yeah, we get sustainable, innovative hop products. They are better for the environment because they have a higher productivity. A new variety has higher productivity, needs less resources, but also on the application side, a new product has yeah, better yields in the brewing industry, and therefore it's better for the whole environment. It's also better for the economic situation because of better utilization of resources. Yeah, you have less costs and a higher income, and this ends up in a stable, yeah, kind of a stable income along the supply chain. So the system is more stable, and in the end, everybody is more happy, starting from the grower, but also to the brewer. And so this were about the global facts and about how do we define science-guided product development. And now I would like to go more into details, and I would like to show you the concept of our so-called Aroma project. In 2020, we started to analyze 150 different genotypes. We did the sensory evaluation with them, but also the chemical analysis. And then what we wanted to come up when we correlate sensory and chemical analysis, that we come up with aroma associated compounds. So which compounds in a hop are responsible for a certain sensory impression? And what we found out is 150 genotypes is already a good number, but it's definitely not enough to, especially when you look into prediction or random forest models. So therefore, we nearly doubled the amount of samples in 2022. So nearly 300 genotypes were then assessed again. We did the sensory evaluation and the chemical analysis and made correlation and ended up with a better set of aroma associated compounds. And last summer and this winter, we evaluated 100 different genotypes in beer, because as I said, what we want to come up is important compounds and their role in beer. And therefore, we screened 100 genotypes in beers. We did also the sensory analysis of them, but I come to that later in my presentation. And what we also do is a genetic analysis to come up with genetic markers for sensory impressions, but also with genetic markers for certain compounds which are causing a certain sensory impression. And how did the sensory actually work? In 2020, we started with 6 categories, 6 defined categories: citrusy, fruity, floral, herbal, spicy, resinous, and onion garlic. And what we found out, what is not citrusy, fruity, resinous, and onion garlic is floral and herbal spicy. And since we had then more genotypes, we skipped the categories floral and herbal spicy in 2022 and just concentrated then with a bigger set of genotypes only on citrusy, fruity, resinous, and onion garlic. For that, we have a panel here in Mainburg consisting of 11 panelists in 2020 and 15 in 2022. We trained them with aroma references for each category, and the evaluation was done for each sample in each category with a 1. So the aroma category and the aroma is present or is recognizable, or the aroma is not present with a 0. And this helped us to quantify sensory impression of hops. The samples we evaluated were hop teas, where we used 200 milliliters of boiling water and 6 grams of hop flowers, hop cones, and made a hop tea. First, as we started in 2020, we wanted to evaluate raw hops as it is, but it was during the pandemic, and so we needed to find a solution where everybody gets the sample and is not in danger because of COVID. So we came up with a hop tea method, and at the end we were really happy because then everybody got every day a box of 20 samples on the desk or even into the home office, and we ended up with a really good data set even during the COVID time. And as you see here, then afterwards, after the sensory evaluation, we checked with multivariate data analysis if we have covered a certain diversity of hop aroma, and I can say yes in 2020 with the 6 categories: onion, resinous, citrusy, fruity, floral, and herbal spicy. You see that the samples are some go into this, they go into different directions. These are the samples which are more resinous and onion garlic. These are the fruity ones, citrusy ones, and all these here are the herbal ones. And as I said earlier, we found out basically what is not citrusy, fruity, resinous, and onion garlic comes through that is kind of herbal. There's a typical hop character, and therefore we skipped that in 2022, and we only defined fruity, citrusy, resinous, and onion garlic, and all these here are not fruity, not citrusy, not resinous, and onion garlic. They are more the herbal ones. And after the sensory, the chemical analysis was performed, and this is done in collaboration with the Julius Kühn Institute, with the JKI in Berlin, in Dr. David Rives' group, and Jana Böttger is the PhD student there, taking care of all the analysis and the aroma correlation. The method is an untargeted analysis of the volatile compounds by SPIMI GCMS, and aqueous extracts were analyzed, and the outcome is now a peak table with nearly 800 detected compounds. And these compounds which were detected were then the basis to make correlations, because we were then able to make correlation between the sensory evaluation and the chemical analysis. And this is here an example of one metabolite, of one compound. And so on the x-axis, you see the abundance of the metabolite, and on the y-axis, the sensory evaluation of that of all these samples in terms of, for example, here onion garlic. And this is a quite nice correlation because the higher the concentration of this compound, the more onion garlic aroma comes through. And this is what we are looking into. In a linear model, we want to see if there are compounds in higher presence causing a certain aroma impression. And once we had this linear regression type done, we started also to look into random forests, so machine learning models and nonlinear models. And what we did is we tried to predict, or what we want to come up is to predict certain aromas when you have a when you see a certain chemical profile. And on the x-axis, you see here predicted sensory expression of onion garlic, and here sensory expression of onion garlic. So the actual sensory expression we screened in the sensory trials, and this gives us also we see a very good correlation here, which tells us this model what we created really gives us so when you have a certain chemical profile and you use that model, the model can tell you is this sample onion garlic or not. And this is also what we do for other categories, but onion garlic here is an example. Yeah. And what you also can do out of this random forest, we can distinguish, or we can investigate the feature importance. So how important, because we got many candidates out of it, but how important are they really? And this gives you the feature importance, tells you how important certain compounds are for a sensory impression. And masses like the ones here I marked, they are really interesting ones. And it seems like they have a huge impact on a certain sensory impression, like here the onion garlic. And what has been done in the past months is also a GCO project at the JKI in Berlin. They screen for certain compounds, and we already found causative candidates. So because this is all based on models, but we also found in that GCO project that some of the interesting masses I show here have a certain impact on the aroma, and we can actually smell that, for example, these compounds are responsible for the onion garlic, for the off flavor. But you also do this for citrusy, for fruity, and so on. And and this really helps us to get a better understanding of what is happening in hops. And some of the masses we detected here, which have an influence, are not known yet. So next step is then also the elucidation of these compounds and to include them in our future development system. And the last step is then the genetic analysis, where we create where we have sequenced all the samples with GBS, and we call the polymorphisms, the single nucleotide polymorphisms, and they are then used in a genome association study to create genetic markers for a certain sensory impression, or to create certain genetic markers for chemical compounds. And this can be then used in the breeding program as a genomic prediction of aroma relevant compounds. And this really helps us to select the best parents for the future. And as I said, hops is dioecious. There's a female and a male plant. We can screen the females for aroma and so on, but there is no chance to screen the males because they don't form flowers. You cannot evaluate the sensory impression or the sensory of these plants, but it gives 50% of the genetics. So these genetic markers can help us to select better males in the future and to make better crosses in terms of a certain aroma impression. And I was talking about the onion garlic aroma as an example. This is a really interesting category for us. We don't want to breed hops which are onion garlic, but we want to breed against. So we want to breed varieties which don't have this kind of aroma, and we use that as an off flavor marker. And in most cases, it is easier to breed against something instead of breeding for something. And to test also the varieties and products, we have a research brewery in Naimburg, 5 hectoliter brew house, and we have different types of and sizes of fermentation tanks. So starting from low volume fermentations to fermenting also a complete batch of 5 hectoliters. So we are very flexible in the brewery, and we can do what we need for terms of bitterness evaluation, or also aroma evaluation. And just recently, we made also the benchtop brewing trials. This is Florian Simpfendorfer's bachelor thesis. He created a certain base beer, and then during dry hopping or during fermentation, all these beers were dry hopped. And as I said earlier, we selected 100 different genotypes for that. They were selected based on sensory impression, but also because of a certain chemical composition or because of a certain genetic background. And so there were 100 different beers were created, and they were then filled also in mini vials and stored at -80. And they got then tasted by the panel in the last few months. So we started in January, when we finished last week. So 100 beers were tasted by 15 panelists in 4 categories. So more than 4,000 beers were tasted in the last months, and I'm super excited to look into these results, because this will actually then answer us more questions in terms of what is potentially in a hop and what ends up in a beer. And once we have done the analysis, we will also the sensory evaluation. We will also then look into the chemical analysis, so what actually ended up in beer. And this comes then again to this slide, what science-guided product development means. So what we screened certain hop genotypes, and we found out what is potentially in a hop. We found out how they smell, the different hops, and what has a sensory impression. And we, as I said, we brewed so many beers out of it, and we tasted that, and and we are getting closer to our ambitious target to identify aroma relevant compounds in beer and hops and their role in beer. And this, as I said, helps us then for making the varieties better, but also making the products better. And now I would like to give you a few examples how this knowledge was applied. And for example, the variety Acquia was released a few years ago to withstand climate change, and Acquia has a very classic aroma characteristics comparable to the variety Perle. And it was developed because certain varieties such as Perle are under pressure because of climate change, and we wanted to come up with a solution which tastes and smells comparable as Perle, but gives more stable yields and also more stable alpha contents. And with Acquia, we found a good candidate, and we did a massive tasting round at Brau in Nuremberg a few years ago, where 315 tasters evaluated beers made with Perle, a beer made with Perle versus a beer made with Acquia. It was a lager beer, and the results are published in Brauwelt International in 2021. If you're curious about the whole setup and more results, and we asked the tasters to compare these beers, and if they find differences between the bitterness quality, bitterness intensity, and the overall impression, and there were no significant differences between the beers. And we also asked for preference. So would you prefer one beer over the other? And there was also no significant differences, which told us these two varieties are comparable, and were then the reason why we launched a little bit later than the variety Acquia. Solero is another example in a different direction. Acquia is very classic. Solero, on the opposite, is a very tropical fruit, mango passion fruit variety, so more for craft application, for hop forward beer. It has very stable yields, very good resistances, but a very unique aroma profile. So that's basically the opposite of Acquia in terms of aroma, but it's also very, very pleasant and gives very good beer results. And just recently, what we released is Helios. So Solero, Acquia, two examples for an aroma variety. Helios, a new high alpha, which combines good resistances and mild aroma. Helios is very resistant. It has 3 resistance alleles against powdery mildew. It's also very downy mildew resistant, has a mild aroma and high alpha and high yield. So Helios is a result of 2 decades of research, because all is combined in this genotype. All the genetic study, the metabolomic studies we performed in the past, everything is kind of combined in this genotype. And on the product side, so not making only varieties, but also making products out of it. In the past, we've created certain liquid aroma products, such as our hop oils, type Noble, which can give the beer a late hop character, or the type Hoptanical. This is hop oil plus a botanical, for example, lemon or ginger. And just recently, we created a new category, type Selected, and there was another hop oil in it for especially in the low alc and no alcoholic beer application. And also Salvo is a new product, was released a few years ago. And all of them, they have really good product advantages, because there's no added vegetative matter, so you don't add plant material to your beer, which reduces wort losses or beer losses. You have unlimited flexibility, because you can use it downstream, certain hop oils, and out of one base beer you can create different kind of beers. It reduces the carbon footprint, because they are very concentrated products, and you don't need to ship as much as with pellets compared to pellets, and also storage is much more limited and lower. And these products have higher yields due to excellent solubility of aroma compounds. And one example I would like to give today about the hop oils, because this was released last year, type Selected Free, is for the application on the non-alcoholic or low alcoholic beers. It gives the beer more body and complexity. It gives improved mouthfeel and a pleasant hop aroma. So this is really a candidate we came up because of studies we made in the past, and what do low alcoholic beers need, what do we have to get back, and what is necessary to give the beer more body and more complexity. And another example I want to give today is the Salvo we have now on the market. It boosts aroma and flavor as a variety-specific aroma extract. Has also no bitterness effect, so only on the aroma side. And also there you don't add vegetative matter, and yeah, reduces wort losses on the late hopping or whirlpool edition side. And initial trials have been shown that Salvo can extend shelf life in most applications. We are currently verifying that, but already preliminary results show that this product is also better for shelf life. And this brings me to the end of my presentation. As I started, my presentation is about global climate change and regulations and so on. So innovation and innovation of new varieties, but also new products, is essential for a successful industry future. And here I'm not only talking about the hop industry, it's also about our common beer industry. Innovation is essential for our future, and new products, new varieties can make the world better. And a better understanding of complex traits like aroma and resistances gives us the possibility to be more precise in our aroma breeding program, but also gives us a better idea what actually needs to end up in a hop product to be applied then on the beer side. And so a better understanding of what is happening in the whole process from the hop to the beer gives us the possibility to make better products. And of course, such new products and varieties have advantages. They are more environmental friendly. They have better yields in the field, but also better utilization in the brewing industry. And all these products and varieties, can give a more stable income along the whole supply chain, from the brewer, from the grower to the brewer. And this is the end of my presentation, and I would be more than happy to answer questions. And thank you very much for listening, and I'm really curious about your questions.
Thank you very much, Alex, for your comprehensive talk. Also compliments from the virtual audience. We have a couple of questions. So the first one is regarding the process of making the hop tea for the internal evaluation. So how long is the hop being boiled in the water, and is this afterwards being cooled down to avoid aroma loss?
That's a very good question. So what we did is we heated up the water to 100 degrees. Then we added the milled hop flowers, and no, it wasn't the milled. That was the whole hop cones. Whole hop cones added, and then we closed the lid so that nothing evaporates and stays in the system. And then it was cooled down. It stayed for 24 hours in that jar, so it cooled off. And the next day, the following day, when it was cooled off, there was then the tea aliquots of that tea filled into the vials and then distributed to the panelists.
Okay.
We did trials in the past where to come up with that method. We compared, of course, raw hop aroma and that tea, because the original idea was that we use raw hop as it is to evaluate, but we were limited on samples and limited on during COVID. So we compared it, and we were really happy about the tea. The tea is a little bit different to the raw hop, but what actually ends up in that tea is the soluble compounds, and which also then end up in beer. So the hop tea of the evaluation is closer to the beer at the end. So it was for us the best method.
Yeah, it makes sense that you have the liquids in the water or beer.
Yeah,
at the
and
end.
also later in the chemical analysis, there was also an aqueous extraction,
Mhm.
and so what we smelled in the tea, in the water based tea, is then also then later on analyzed, and then we could make the sensory the correlation between that tea and the aqueous extraction on the GC.
That's good. It's a good point. You mentioned the GCO. Maybe you can just give us some detailed, or for someone who's not familiar with this, what is it and what can we do with the GCO?
Yeah. Yeah, good. So GCO means gas chromatography olfactory analysis. So you have a GC, a gas chromatography machine with a sniff port, and so a sample is injected, and then during the measurement, you can smell what is currently analyzed, and when you smell that, you find different aromas. You find, oh, this is very rose-like, and so, oh yeah, this could be geraniol, or this is very citrusy, floral. This is more like linalool, and but also you smell off flavors and very onion, garlicky, or very moldy character. And this helped us really to find, okay, we know that certain compounds are in a certain area, have a certain retention time, and in that window you can really check, is this really causative, and is this really that aroma impression we want to see? And yes, we could verify certain compounds by that.
Yeah, that's good. Thank you. So there are some other questions. So also the question, I think it's about the hop tea. Did you adjust the pH?
No, we did not adjust the pH. It was just a pure water.
Okay. So so another question is also, where do you see the limitations in detecting the aroma relevant compounds?
Pardon?
Where do you see the limitations in detecting aroma relevant compounds?
Yeah, the limits are really limitation is that sensory is a very, very complex trait, and it has there are synergistic and additive effects. So not really everything can be, especially in the linear modeling. So the linear model is based on the more of a compound is present, the higher the sensory impression. This works for certain compounds, but certain aroma impressions are built because of a combination of different compounds.
Yeah.
And a special fruity character is because of a combination of certain esters in a certain concentration. And because basically all compounds are always present, but it always depends on how much of them are present. And this is really limiting the linear model, and that's why we use also machine learning models and nonlinear models to find out that synergistic effect. But this is really the challenge, and AI, artificial intelligence, is helping. It gives us a better understanding and in such models, and this is something where we look into in the future to use more artificial intelligence for finding out important sensory compounds.
So because of the complexity.
The complexity.
Yeah. So, okay.
This is one,
yeah. So there's a question regarding the new product. So you mentioned that this product can enhance the body and improve even the shelf life. The shelf life can be improved in which point of view? The microbiological, or?
This is two different types of product here. So the one product is the selected type 3, which was exclusively, or it was specially developed for the non-alcoholic beers or low alcoholic beers. the compounds, so the monoterpene alcohols, which are enriched in this product, they can help to make a better to improve mouthfeel and body. And that's also known in literature that certain alcohols can improve that whole complexity and of that beer, and gives the beer more complexity and therefore more mouthfeel and a more pleasant taste. On the shelf lifetime, this is, for example, the Salvo I mentioned, because of adding no vegetative matter or no plant material, which can then form, which also brings iron in the product. The beer is then not that stable, but with adding no plant material into the beer, the system is then also more stable, and it has nothing to do, I would say, with microbiology. It's more like oxidation of the product.
Okay.
But at the end, adding no material is positive on the one side, because it makes the beer more stable. But on the other side, also polyphenols are not added, and this gives also then a different character to that. So it is always what you need, what your target is.
So you can use the polyphenols as antioxidants in your product also. Yeah. Okay, thank you. Maybe last one question. You mentioned Helios as a new variety. Which beer style prefer Helios, if I can say it like that?
Helios is, as I said, a high alpha, and major applications on the bittering side.
Okay.
And but it gives a very pleasant bitterness compared to varieties which are existing in the market. The main application is on the bittering side.
Okay.
There's
more question.
Okay, here. This one.
So liquid aroma products need a carrier to add to beer, on how they must appear in labeling are the questions we have here.
Some of them are diluted, or the carrier, the solvent is propylene glycol, and some of them, it depends on the regulation where you are. So this question is really like, it depends on in which country are you producing. So there is for certain compounds there needs to be labeled, but propylene glycol does not need to be labeled, and also in the EU, depending on how much is in the product, so they have a certain limit, but this usually goes not with our products. So with the products, they don't need to be labeled in certain areas. But if you're interested really in these products, and you can always contact us, and we have colleagues here which can absolutely answer your question for your area, and what kind of beer and what kind of product. So there is not a simple answer to that question. It really depends on, are you talking about an oil? Are you talking about an extract like Salvo? There is no solvent used, so it's basically the hop extract, fractionated. It really depends on where you are producing your beer and what kind of liquid product you are using. But if you're interested in these things, please contact us, and we can give you then a clear answer to your question.
So another question is the onion garlic aroma. Is it positive or negative to beer? I guess you mentioned it before. It is as a not positive. So just to avoid.
I was talking about this onion garlic flavor because for us it's an off flavor, of course, and nobody wants the beer to be that way. But it gives us a good example of how the whole aroma study works, and to better understand what is onion garlic, because this is not really well investigated. Everybody was in the past was focusing on the good aroma, on floral, herbal, but also fruity and citrusy compounds. But the bad ones, the bad guys, haven't been studied in that deeply. And this was why we are really interested in, because at the end, what doesn't smell bad smells good.
Okay.
And this is also known in other crops, or executed in other crops, breeding against something. So breeding against certain off flavors. So breeding, that these compounds don't show up anymore.
To avoid the special.
Yeah, this is a really good question, and nobody wants a beer like that. Probably some do, but
we don't know.
but our main target is to breed against that specific type.
Okay. And the question about the beers which were evaluated, so base beer was a lager or ale, and do you consider biotransformation changes?
Yes. The beer is an ale type, was a top fermented beer using the US-05 yeast, so very neutral, a neutral yeast. But yes, and there is always biotransformation. And we had to decide for one system and for one setup. We made also trials. We added the hops at the beginning of fermentation, but we also tried it before at the end of fermentation, or even some whirlpool beers, and we ended up, the best method is with a very neutral yeast at the beginning of fermentation to get a certain profile out of it. But of course, that's one setup. We can make assumptions out of that setup. We cannot say generally this is covering all beer applications. This is like, okay, we have this in hops, and this, because of that setup, ended up in beer. But this doesn't really mean that we covered everything, but this is the first attempt. Next would be then whirlpool application and so on. But we tried it, and the best results for our initial trials were beginning fermentation, adding the hops.
And you have the same procedure for all your genotypes used in this trial.
It gives the best profile of a hop in a beer. And but yes, we already see we see major differences between raw hop and beer. It is a different system, but it will be really interesting to see what are the differences and why certain compounds are not showing up in beer anymore. Is it because they were evaporated, or they were transformed, and so on? So it's going to be a really interesting result, and also gives definitely more questions for the future.
Of course, yes. So there's also a wish to decrease the production cost of Salvo. I guess this is just a comment.
Yeah, it really depends on, especially Salvo is variety specific. It really depends on which variety you are looking into. So there's and comparable to the price of when you add that amount of product to your beer, or you want a certain aroma profile, and you add this variety as a pellet and as a Salvo, you have definitely a better pricing than compared to what you have with a pellet. And really depending on the variety, there's what goes into that is a huge difference. But at the end, what we try is not to make our products, so make comparable products to pellet pricing, and not create more cost-intensive products.
Okay. So I think we answered all the questions so far. So there's.
I think yes.
Okay, then I would say it's time to close the session for today. So thank you again, and thank you for participating. And if you are interested in on more informations about the topic, you can visit also our hopsteiner.com website and check the section technical publication. After this webinar, you will receive an email including a link to watch the recorded video of today's session. And again, thank you for joining our Hopsteiner Forum. And we hope you enjoyed the time with us today and looking forward to meeting you soon. So thank you and goodbye.
Thank you. Bye.